Yeah, I'm going to use this because I like to move around a little bit on stage. So hi everyone, I'm Shay. I just want to start off by saying thank you to the organizers for the entire conference. I think it's been fabulous, and for inviting me to this session and to talk about things that associate with plants. So it's kind of great. I'm a graduate student at the University of California, Merced, and I am here with this session to talk about plant-biotic interaction. So plants, they are not alone. They're out there collaborating with and fighting with other organisms all the time. And so this is just going to be like a snapshot of some of these interactions and, how they may be helping plants or, antagonizing plants or whatever. And it's just a great opportunity to kind of think a little bit more broadly about these community dynamics. And I think it's really great as, plant botanists and biologists to really make sure we're considering it a holistic view of things when we look at it. I have a background in mycology, so surprisingly I'm not a plant person, but I hung out with a lot of botanists at San Francisco State, shout out to all them. And I think they rubbed off on me because I study plants now. So I'm going to talk to you a little bit today about my dissertation work on endophytes and monkeyflowers. Okay, I'm going to start off the talk with the thank yous because I don't want to rush that at the end. I want to thank my advisors, Jason Sexton, who's here right there, hi Jay, and Caroline Frank. And then my fieldwork and support, I want to give a special thanks to Lily Pennington, who's also right here, thank you. She is in like 2 of these pictures and did a lot of the field work with me, as well as Jasmine and Stephanie, my undergrads, and that's my husband hiking along the PCT just in the red meadows outside of Mammoth Lakes. And the cooler that he's holding, Shenjiao mentioned her car, and I want to mention my cooler. Special thanks to my igloo, because that thing came with me all over the Sierras, and I dropped it down a cliff a couple times, and it survived, so Igloo do not pay me to say this, but I just thought I'd share it with you. Thank you to Northern California botanists and all these other funders for contributing. Societies are the best, right? Okay. So just a quick overview of what I'm going to talk about. I'm going to introduce you to endophytes and just plant microbiomes in general. And then I'm going to go over 2 experiments that I'm doing, a drought experiment and the field experiment that I just spent all last year doing. And then hopefully we'll have time to look at some really beautiful pictures. So as you gaze at this little mixed collection of monkeyflowers looking at Mammoth Mountain, I just want to introduce you to microbiomes. So as far as we know, all multicellular organisms have a microbiome. You all have a microbiome on your skin, in your gut, in your eyes. Your cats and dogs have microbiomes, and all plants have microbiomes. And what we know is that plant microbiomes are an extension of the plant phenotype. And so when we're thinking about plants and we're thinking about how they're interacting with their environment, we should be thinking about these communities of microorganisms along with that. So where do these microbes live? Where do they come from? Well, they come from everywhere. But they can come from the soil where the plants can uptake them into their roots. They can enter in through the stomata. They can come from pollinators who are coming to visit their flowers and deposit them into their little, into the canals of nectar. And we're going to hear a little bit about nectar next with Callie, so a little shout out to her. And they also get passed down, like our microbiomes get passed down by our moms and then change where we live and eat. Their microbiomes also get passed down by their parents and their seeds. And so plants don't move around like we do, but they are impacted by all these other factors that can change their microbiomes. I focus my attention on the microbes that live inside the plant. So endophyte, inside plant. These are the ones that live their whole life inside the plant, and sometimes they can come from the outside and reproduce in the plant and stay there forever. And that's really cool. So that's what I look at. And the reason why I study endophytes is because we know that they help plants grow, they help with plant growth, and they also help plants respond to stress. And that's my area of interest. I'm interested in stress and what plants are going through and how they're coping with stress. And so here we have a study done by Rusty Rodriguez in 2008 where we have some rice on the top and tomatoes on the bottom. And on the left-hand side, they're dealing with drought and saline solutions, so salinity. And so on the left-hand side, we have plants without their endophyte communities. And on the right, we have plants with their endophyte communities. And so you can see a clear distinction in the plant's ability to cope with that stress that it's dealing with in that environment. And what's really important to remember is that when we're thinking about endophytes, we're thinking about the composition of the community. And that's really what I look at. It's who's in that community. It's not just one microbe or another microbe. It's really the collection of microbes that makes the difference. And so when I'm thinking about endophytes, I'm thinking about it on a whole community level, and I think about the composition of the makeup of that community. But we still know, it's really great that this stuff happens in agricultural plants a lot, but we still know very little about how these endophytes are composed and what that means in the natural world. But we know that the natural world is experiencing a lot of stress. Climate change is having a huge stressful impact on populations all over the world. And so I ask, is it possible that endophytes play a role in mitigating this climate stress in natural populations? Are they doing anything to help plants cope with this rapid change? So my main kind of starter question, just to get going, is how do these endophyte communities respond to stress? And when I was starting my project and thinking, where do I start here? I was trying to think, which plant am I going to work on? And I work with Jason Sexton, so that kind of gives you an idea of where I got started thinking about it. But there's some things I needed. I needed a plant that was well studied. I'm not trying to reinvent the wheel here. I needed something that was in a natural system, hopefully locally, something we knew that was going through climate stress, reasonable range size, and ease of manipulation. So, monkeyflowers. Naomi did a really great job yesterday of telling us all about the wonders of monkeyflowers. They're diverse, they're highly studied for ecology and evolution, they're really well understood in their genetics, they have really unique mating systems, and so when picking monkeyflowers, it was a surefire bet, but there's so many to pick from. So when we kind of narrowed down a little bit more what we wanted, the cutleaf monkeyflower came out as the winner for this project because the cutleaf monkeyflower is an endemic species of Sierra Nevada. It only occurs on the western slopes of the Sierras. It's got a very unique ecology where it grows in these sort of mossy patches sometimes, most of the time, on granite rock outcrops. And so this is a really harsh environment. There's not a lot of competition. And they've sort of found this really unique niche that they occupy. Sometimes they grow in the cracks of granite, like that picture on the left. And we know that they live in this stressful habitat. They're pretty easy to collect and grow. They have their little capsules held with their seeds, take me, study me. And they grow very fast, so in a lab environment, we can grow them up in a couple of weeks. And they're annual plants, so we can measure something like lifetime fitness, which is a pretty powerful thing. Looking at their entire species range, they kind of have these different climate habitat types across this elevation gradient, which is really nice sort of natural treatment, where they have a low elevation population composed primarily of chaparral. Temperatures there are pretty warm, and it experiences a lot of low precipitation and drought. The central mixed Sierran forest is more of a kind of cushy environment for them most of the time. And then the high-edge subalpine population has its own stressors, with it being extremely cold and sometimes even having low precipitation there. And so here we have the Sierra Nevada range in this little thing, and here's Mono Lake. This is probably Mount Dana kind of representation, and then here's Merced in the valley. So we're nearby, which is cool. And so what we did for this first project, just to see if these communities change in response to stress, was we collected soil from the middle of the species range, because we didn't want it to be necessarily adapted to any of these conditions. But we wanted to see if we apply stress, do these communities shift. And so we collected this soil, we mixed it up in a big pot, homogenized it so it was all consistent, and then we just planted that soil into 3 different trays with 36 wells and let the seeds that were just naturally in the seed bank grow up. and then we weeded out whatever seeds were not, or whatever plants were not supposed to be there when they did grow up. And then once they grew up, we put them into 3 treatment groups. One of the treatment groups was like a pre-harvest group, which meant I took all the plants out before starting the treatment. And then the 2 groups that were left was the control and the drought. The control just kept getting water, and the drought stopped getting water and dried out. And that lasted for 2 weeks. So the pre-treatment group was right when they grew up, collected them. 2 weeks later was the treatment with the control and the drought. And so those are our 3 groups. And so you might be wondering, well, how do you just get the endophytes, right? There's all these microbes everywhere, you just told us that, so how are you isolating endophytes? It's a pretty cool technique. This is what the plants look like when I harvest them. They're pretty small. This is in the palm of my hand with a white glove. I probably should put my hand in there so you can see it for scale, but they're pretty tiny. And there's their shoots and roots. And I basically take the plant, I clean up all the soil, and I put it in a tube with some buffer, and then I put it in a sonicator, which uses like sonar to shake the plant with vibrations from the sound, and it shakes off all the microbes on the outside and just leaves the microbes left on the inside, which is pretty cool. And so then, I do some next generation sequencing. I use ITS for the fungi and 16S for the bacteria. Used aluminum-iSeq at the Joint Genome Institute and CHIME 2 to analyze the data. And basically what we see is a pretty cool result. So for species richness, which is just presence or absence of microbes, for both fungi and bacteria, the root has a much higher diversity. There is just more of these microorganisms living in the root than the shoots. Not a very diverse group in the shoot. And that's kind of expected, right? Because they're getting a lot of their probably community from the soil or interacting with the soil in some way. We didn't sterilize the soil, right? This is the native microbiome population we collected with the soil. The water we gave them was sterile, so we're trying not to have outside influence. When we tease apart the treatments of the group, we see that the shoot community here in green, this is the principal component, so the distance between, the closer the points are together, the more similar, and each point represents an entire community of microorganisms for one plant. And so with the shoot community, it didn't really change that much between treatments. The root community changed pretty dramatically. It changed between the different control and drought, and it changed with time. So that 2-week difference in the pre-harvest and the treatments actually made a difference in the community. And the same is true for fungi. That's my statistics in case you need it. So for this first project, when we asked this question, drought does alter the root community pretty significantly. And both the composition and species richness were increased in the root more than in the shoot. And lastly, this root community, may be playing an important role in drought stress response. And so now that we know that the communities of this plant, the Endophyte communities of this plant, are changing with abiotic factors, we want to test this out in the field. We know that the plant has this natural elevation, climate gradient treatment, and so we asked, do endophyte communities change in response to climate variation across an entire species range? And this is the first time something like this is being asked in a native population across its entire range, looking at species variation in relation to climate. So it's a pretty cool question, and I'm really excited about it. We also know that the plant is experiencing variation in fitness in response to this climate. Here we have 3 populations that are originating from a high elevation cold environment, and 3 populations originating from a low elevation warm environment, and a common garden experiment where we have a cold garden in the high elevation and a warm garden in the low elevation. And when we plant those plants in those 2 gardens, we see this range of variation of fitness in the cold garden and reduced and less varied fitness in the low garden, which suggests that this population's fitness is variable depending on that climate. So we know that the abiotic factors are affecting the communities, and we know that they're affecting the plant. So let's put them together. So last year, I spent the whole year going out and sampling these plants through the field. So I was collecting from April to September. We did 3 sites in warm, 3 in the central, and 3 in the high. 30 plants per site. I'm testing 4 compartments, so the root and the shoot like we did before. and the soil and the moss as well to get any sort of environmental variation. And so it's 27 trips. It took me all year last year, 270 plants, and then a little over 1,000 samples when you include all those compartments. And so we're testing for all these variables, wind direction, speed, soil type, population, a lot of metadata. Basically, I expect to see some microbial communities of these to be shaped by climate. That's what I'm expecting. I hope that, what we're going to see is that in the low elevation, there's going to be microbes that are only associated with the low elevation in all 3 sites, and the same for high. And I expect particular taxa to be more abundant in some sites. So for instance, more abundant, like one taxa would be more abundant in the low elevation than in the high, suggesting that it might have some role in helping the plant adapt to that climate condition. And so up next, I'm working on sequencing and identifying plant genotypes using microsatellite data and doing some indicator species analysis to pull out this taxa information. And I really quickly want to share with you, because I've crunched into time and questions, just some pictures of the habitats. So this is a low elevation chaparral of the backyard of somebody's house in Oakhurst. This is at Hetch Hetchy, where the monkeyflower actually grow out of sedum, which is pretty cool. This is off of Highway 168, really cool demonstration of how they just kind of pop out of these cracks. So that's them growing up under the crack there, this big Granite Cliff. This is right off of Wawona Tunnel in Yosemite. When you go up to Wawona Tunnel View, there's a big rock outcrop right there. You can just park your car at the parking lot, ignore all the people staring at you, and these are the monkey flowers right there in the rock, and it's a pretty nice view to sit and sample for the day. Tenaya Lake, if you go further into Yosemite, and then Grand Bluffs here. This is my advisor Jay. Another shout out to him. He's contemplating his love of monkey flowers, looking across the South Sierra forest. And thank you very much for your attention.